Okay, so I would like you to try to complete this by filling in the blank. Codons are made from blank nucleotide base pairs of RNA and code for what? Pause here, try to fill in the blanks, and then we'll go over the answers. If you said that codons are made from 3 nucleotide base pairs of RNA and code for amino acids, you would be correct. As you can see, there are 7 different codons here in this image, and they each code for a different amino acid. That is, except for the stop codon, which tells the ribosome to stop creating the amino acid chain. Now, as I said before, today we're focusing on mutations. So hopefully there are some things that you remember about mutations. You might remember from our previous classes that mutations are any unpredictable change in the structure or amount of DNA in an organism. Most mutations, something that you might not already know, is that most mutations do occur in your normal body cells. Okay, so they're not really going to be passed from one generation to the next. You may have had some mutations happen in your body already throughout your lifetime. Many humans do, but that doesn't necessarily mean that you're going to be passing that on to the next generation. Only the mutations that occur during formation of gametes, so during the formation of egg or sperm cells, only those mutations can actually be inherited to the next generation. And then once that generation does have the mutation in all of their cells, including their gametes and body cells, then that can further be passed on. So for example, if you are lactose intolerant, it means that somewhere way far back in your family history, there was a mutation that caused your body to not produce the protein known as lactase. Okay, so that means that a mutation occurred in the DNA or RNA in one of your ancestors' bodies, okay, and specifically though in the formation of their gametes, and then that mutation in the code made it so that their bodies did not properly produce the protein known as lactase. Okay, so if you are lactose intolerant, you don't have the protein called lactase. Lactase helps you to break down dairy products so that you can process it and break that down properly. The next thing I would like you to do is take a look at this GIF about gene mutations. These are the 3 different types of gene mutations that you need to know about. So first, just pause the video here, and I want you to be seeing, thinking, and wondering about this GIF. So you want to make some observations first, then make some inferences about what you think is actually happening there, and then come up with a question based off what you're observing. Pause here, and then we'll talk about some of these observations, inferences, and questions that you may have come up with. You may have noticed that during an insertion, this is a type of mutation where a base is being inserted, a nitrogenous base is being inserted. We have an extra adenine being inserted between the C and the T. In the deletion, we have the deletion of a thymine from between the C and the G. It's being removed. So their names, this is kind of nice because for these mutations, their names sound exactly like what is happening there. Okay, for substitution, you can see that the thymine is turning into the guanine. And so this is where one base is being substituted or replaced with another base. The inferences that you might have here are that the codons, how do you think the codons might be affected by the insertion, deletion, and substitution? Pause for a moment and think about that. If you said that the codons might change, you would be correct. There is a big difference, though, between the codons changing in an insertion and deletion and the codons changing in a substitution, and that's what we're going to talk about next. your question might be, how does this affect my traits or my proteins? And so that's a great question as well. And again, this kind of comes down to what we were talking about before about the codons changing. How is that actually going to affect your traits and your proteins? Let's go over substitutions first. Please take notes as I'm completing this. For a substitution, remember, we are replacing one base on the RNA or DNA with another base. So in this case, we're going to replace the A that is found right here with a U. So we are substituting the A for the U. This is why it's called a substitution. Let's take a look at what ends up happening there. If we look at our amino acid chart, we can see the original codon was GAG. So how do we find out what the amino acid is for that codon? Look over here at the first letter in the codon, G, then A, the second letter, and then the third letter, which is going to be G. And we can see that it's GLU is the amino acid, right? The amino acid is GLU. Glutamate. Now, will it still be glutamate once we've replaced the A with the U? Let's see. Now we have GUG. So G, U, G, and we actually now end up with VAL, or valine. So no longer do we have glutamate, we now have valine. How is this going to affect the structure of the protein? If you said that it's going to change the structure of the protein, which could also make this into a different protein, you would be correct. Now let's take a look at insertions and deletions. Let's say that over here at GAG, we actually inserted a letter right here. Okay, so now we're going to actually insert a U instead of replacing the A. Okay, so instead of replacing the A before, we replaced this A. So we got rid of the A and we put in a U in its place. But over here in the insertion, we're actually just going to add a U right here. Now what this does is something very significant. It's called a frameshift. This is called a frameshift mutation. Okay, this actually takes the frame of all of the codons and shifts them over to the right direction. What do I mean by this? Well, now we've reordered all of the codons. So it's GUA, GCU, UCG, GAG, and CUA. Now let's find out how this affects the amino acids. So we already know that, okay, GUA is going to end up being what? Let's see. GUA. So G, U, and then we've got A. That is actually going to end up being valine. So let's go back there. This is no longer going to be glutamate. This is now valine, or VAL. How about this next codon? Codon 4 has changed to GCU. It used to be CUU, now it's GCU. So GCU, let's take a look at what that's going to be. G, and then C, and then U. We're going to end up with, over here, GCU, alanine. So we have changed, have we changed this one? Yes, we've changed it from leucine to alanine, ALA. And so on, and you could keep going. So let's see, UCG. U, C, and then G. And we've got UCG is serine. So go over here. So arginine turns into serine. How about the next one, GAG? What would that end up being? G, A, and then G is going to end up being GLU. And then we have CUA. CUA is going to be leucine. Okay, now notice how we no longer have a stop codon there. How is that going to end up affecting our end product? If you said that you think it would just keep on coding for more and more amino acids, then you would be correct. This is going to significantly alter the structure of the protein. So it significantly changes protein structure because it's a frameshift mutation versus the substitution. It only changed one of these amino acids. This is known as a point mutation. Okay, so smaller, or sometimes there's no effect at all, because you can sometimes replace a letter and it still codes for the same amino acid. What do I mean by this? Well, for example, if the G in this GAG codon was replaced with an A, if you look at your amino acid chart, you'll see that it still codes for glutamate. So in that case, if that point mutation occurred where just the G got replaced with an A, it actually still codes for the same amino acid. So this would cause no change at all to the structure of the protein. Now for deletions, it's similar to insertion, except instead of a base being added or inserted, we're actually just deleting a base. So for example, we would delete this A, and now our new codon would be GGC. And then the next codon is UUC, and then GGA, GCU, and so on. This causes a similar change, but in the opposite direction. This is still called a frameshift mutation. Okay, I hope that this video was helpful for you. And as a final activity to help you practice this information, first of all, there are some notes here if you would like to take those. And as a final activity, please find the assignment called Mutations Worksheet on PSL. Complete this activity by Thursday afternoon. So I will give you some more time in class by the end of the day on Thursday submit that to PSL. Thanks so much and have a great week.